Good morning, everybody. COVID-19 conversations are brought to you by the African Alliance, with support from the South African Medical Research Council and the Department of Science and Innovation, and the Vaccine Advocacy Resource Group, which is 100% funded by activists. We bring you these calls in partnership with the Community Constituency COVID-19 Front, the Treatment Action Campaign, and APHA. Thank you for joining us. These calls take place every Thursday at 10 a.m. and seek to bridge the divide between civil society and those who hold power and platform in the COVID-19 space. All of our recordings, along with the transcript and the Q&A sessions, can be found at africanalliance.org.za. We encourage pre-submitted questions and welcome questions or comments in the chat box during the call, and we will do our best to answer all of them. Those we do not get to, we will reach out to our guests after the call to secure those answers, and those will be included in the transcript. Last week, Johnson & Johnson announced efficacy and safety data from the phase 3 ENSEMBLE clinical trial that took place in the US, Latin America, and South Africa, demonstrating its investigational single-dose COVID-19 vaccine that met all primary and secondary endpoints. The Desmond Tutu Health Foundation led and participated in that trial, enrolled just over 400 participants across 3 different sites by the end of 2020. And today, we have yet another vaccine in our COVID-19 toolbox that shows efficacy. One of the first trials that looked at disease and death prevention as an endpoint, 72% effective in the US, 66% overall at preventing moderate to severe COVID-19 at 28 days after vaccination, 85% effective overall in preventing severe disease, and complete protection against COVID-19 hospitalization and death at 1 month, even more effective than the flu vaccine that we all know and some of us have taken. Today, we speak to Gonasagrie Nair, who serves as both Clinical Research Site Leader and Senior Medical Officer for the Desmond Tutu HIV Center's Emavundleni Research Site in Crossroads. Dr. Nair is an experienced public health specialist and clinical trialist. As a younger public health sector clinician in KwaZulu-Natal, the epicenter of the South African epidemic, Dr. Nair was able to appreciate the devastating impact of the HIV and TB epidemic. She subsequently shifted her focus to public health and has over 18 years of research experience, ranging from protocol development to implementation, and is experienced in conducting all phases of clinical trials. These include HIV prevention and treatment trials, vaccines, microbicides, PrEP, HPV, and TB studies. She has served on 2 South African institutional biomedical ethics committees and has been the protocol chair of 2 HIV prevention studies. Dr. Nair is hands-on in directly overseeing all study procedures and management issues at Clinical Research Site, from protocol development to community engagement. Also joining us is Dr. Catherine Gill, who's an experienced HIV clinician and research physician. Dr. Gill serves as both Clinical Research Site Leader and Senior Medical Officer for the Masiphumelele Clinical Research Site, the site of the University of Cape Town Clinical Trials Unit. She has an interest in HIV prevention and adolescent health services and is currently working on a number of vaccine studies for HIV, COVID-19, and HPV. She's currently completing a master's in public health at the University of Cape Town. Drs. Nair and Gill, thank you for taking the time to speak with us, and welcome. Firstly, a huge congratulations, not an easy task, and what a testament to the participants, the communities, and to the researchers. I'll hand over to you now to share with us some context of the trial, the interim results, and walk us through the next steps in ensuring that the money, the goodwill, the time, and the other investments that have been put into this vaccine results in equitable and accessible COVID-19 vaccinations for all who want and need them. Welcome again, and over to you.
Thank you, Tian.
Thanks very much, Tian. Lulu, you going first?
No, you are, Catherine.
Okay, cool. Okay, sorry. I just want to confirm that you can see my my slides.
We can indeed.
Okay, great, thanks. So, hi everybody. It's really great to be here and to be with you and to share our experiences on the ENSEMBLE trial. Thanks for the great introduction. I am a doctor and I'm an investigator at the Masi site, which is in the deep south of Cape Town. So basically what I'm going to do today is to give you an overview of the Janssen ENSEMBLE study. So it was a randomized double-blind placebo-controlled phase 3 study And what that means is that you had an equal chance, a 50-50 chance of being allocated to either the vaccine arm or the placebo arm. Double blind means that the investigator and the patients didn't know which arm they were in. And it was placebo controlled with a salt water injection. And we were looking at the safety and the efficacy of the Johnson & Johnson Ad26 COVID vaccine. So the rationale for the study was to develop a vaccine for coronavirus, and we all know that the world has basically been held hostage by this virus for the last year, and having a vaccine will be critical to containing the current outbreak and preventing future outbreaks. At the time when the study was conceived, there was no vaccine currently available, and we obviously know now that there are a number of vaccines, which is very exciting. So this vaccine is monovalent. It's composed of recombination, replication incompetent adenovirus type 26. It's administered as a single dose, and it's not yet approved. And I'm going to, if this totally sounds like Greek to you, I'm going to unpack that a little bit more in subsequent slides. But basically, monovalent means that the vaccine is only effective against one type of microorganism. And we're going to unpack the other stuff in the next slide. So just to remind everybody, this in red is a human cell. In yellow is the coronavirus. You can see that it's surrounded by like a halo of spikes. On the right, you can see that scientists have mapped that spike, and that spike is now the target for all of the vaccine development. So we're going to see more about that spike later. So this vaccine is an adenovirus vaccine. It's called a viral vector vaccine. There are two main components. That's the adenovirus, which is the vector, and a copy of the spike protein, which is an insert. And a way to remember this is an analogy using a package that you want delivered. What's in the package is really important, but the package will just sit and it won't deliver itself. So we need a truck or something to deliver the package. And what the viral vector is the FedEx truck that's going to deliver the package, which is our viral insert. So the vaccine in this study uses a non-functional version of a type of cold virus, an adenovirus. It's called adenovirus 26. So we've all seen adenoviruses before. They cause colds, diarrhea, conjunctivitis. But what scientists have done is that they have changed the adenovirus to make it weaker so that it can't actually cause you to get a cold. And all it does is it's basically the FedEx truck that serves to get the insert, which is the copy of the spike protein, into the cells. So as I've said before, adenoviruses are common, and this can be problematic because you don't want to show something to the body that it's already seen, because there's a chance that it might neutralize it before it's even had a chance to work. So scientists had to find an adenovirus that was rare. So they've used so J&J has used the Ad26. What other people have used are adenoviruses that infect other primates, like the chimp adenovirus in AstraZeneca. So the AstraZeneca vaccine that's just arrived in South Africa this week uses a chimp adenovirus, but otherwise it's very similar to the J&J vaccine. So then we know you get an immune response to both the vector, so to both the adenovirus and the insert. So it's like a double shot. What's really important to remember, and what I've heard quite a lot of, are people worried that the vaccines actually integrate with our DNA. I think just to say that they definitely don't. They don't go into our cells and then join with our own DNA. Another thing to remember is that these viral vector vaccines are more stable than the mRNA vaccines, and they don't have such demanding cold chain requirements. But otherwise, they are quite similar to the Pfizer and Moderna vaccine. And then if we just look at the study design of this, it was a multi-center phase 3 double-blind. The trial took place in the United States, in South America, in Brazil, Peru, Chile, and 3 other countries, and then of course also in South Africa. And what's important about this study is that we didn't enroll any children, so you had to be 18 and older to join the protocol, but there wasn't an upper limit of age, so it was just above 18. And the other thing that J&J asked us to do was to prioritize older people. So you'll see later in the study results, we have, I think it was about 30% of the older population above 60, which is important because these are obviously the people who are most at risk. So then just quickly to go through the study timeline, the trial is going to last for 2 years. We could screen and enroll people on the same day. They have a visit a month after their vaccination, then after another 2 months, then it's 6 months, 1 year, and then 2 visits in the second year. And what we're doing during that time is following all the participants up to see whether they develop COVID-19 or not. So initially we plan to enroll 60,000 adults to be randomized 1-to-1 into 2 groups. There was also extra information that was collected on some of the participants, some more immunological information, and then a safety subset. The other way the study was designed to increase safety was that we started off just enrolling younger people, so people under the age of 60 who were healthy. Once we had 2,000 of those participants, the vaccination was paused, so the whole study was stopped, and an independent safety committee then looked at that data. When that data was fine, we proceeded then to enroll people under the age of 60 who had comorbidities like obesity, diabetes, and hypertension. And then you can see there was a parallel process. So same thing happened. We recruited people above the age of 60. And after 2,000 participants, vaccination was again paused to look at safety data. And then we started enrolling people older than 60 with comorbidities. So then just to look at the study objectives, the way that COVID-19 was defined was that it had to be molecularly confirmed with a PCR. And then just to note that the severity of all the cases were assessed independently because they were divided into mild, moderate, and severe. The objectives was to demonstrate the efficacy and the safety of the vaccine, and our primary endpoint was occurrence of COVID-19 at least 14 days post-vaccination. So that's the primary endpoint to keep in mind when you're looking at the results. So there were secondary objectives, and those. So one of them was to look at whether we could prevent COVID-19 infection in adults, regardless of their serostatus. So that means we enrolled adults who had previously had COVID-19, and we wanted to see whether we could prevent COVID-19 from happening for the first time or prevent it from happening the second time in those who had already had COVID-19. We also wanted to see, another objective was to see how quickly the vaccine would start working. So we were looking at how cases accrued after day 1. And then we were also looking at whether the vaccine could change how people needed to access medical intervention. and whether the viral load was decreased in people who had vaccine if they got COVID-19. Other things we wanted to look at was what effect the vaccine had on mild COVID, what the vaccine had on what effect the vaccine had on symptomatic COVID, and then also whether we could prevent asymptomatic infections with the vaccine. The study had quite a few inclusion and exclusion criteria, so I'm only going to highlight the main ones. So as I've said, this was a study for adults. We didn't enroll any younger people in our study, and there was no upper limit to age. We didn't enroll pregnant women, but it was okay to be breastfeeding in the study. Then we excluded people who arrived at the clinical trial site sick, so we didn't want to enroll anybody who was acutely infected with COVID-19. We also didn't want anybody who had previously had a coronavirus vaccine. And then the other big thing about our study is that we did enroll people with HIV. The HIV did have to be well controlled, and that was defined as having a CD4 count above 350 and a suppressed viral load. And I know some of the other studies excluded people with HIV, so this study will be particularly interesting because so many of our participants had HIV. And then you were allowed to join the study if you had comorbidities like diabetes and hypertension, but it was quite strict. They did have to be well controlled. The way we managed COVID-19, which was very new for us, this process. So all participants have to complete an e-diary, so either on their phones or on computers, and this provided quite a lot of logistical challenges that I think Lulu might discuss later. So everybody had to have a functioning phone. We had to provide data for the participants, and they have to complete their diaries twice a week for the first year, and then once every 2 weeks for the second year. And that, to be honest, has provided us with quite a few challenges. So the participant is prompted just to say, are you okay? Are you feeling well? Or are you not? So if they are well, the diary closes. But if they're sick, it then starts a whole cascade of procedures, including they have to give all their symptoms, but then we also send participants home with saturation probes, thermometers, and swabs. So they have to swab themselves if they feel sick, and then we follow up with them with another swab after a few days. So then the analyses, the primary analysis has been done. Those were the results that were released last week. There will be another analysis after a year, and then the end of study analysis when all the participants have completed their 2 years. Then just a quick summary of how the study happened. On the 18th of September, the FDA gave us the green light to start, and by the 21st of September, we had our first participant vaccinated in the States. On the 6th of October, the first participant was vaccinated in South America. And on the 6th of November, the first participant was vaccinated in South Africa. Enrollment closed for us and for South America on the 9th of December. So I think we just need to take a moment to appreciate that we recruited 6,500 participants in a month, which is pretty unprecedented for us. And also just to note that the protocol was amended mid-recruitment to decrease the sample size. And that was because J&J ended up cutting 20,000 participants from the study because there was so much COVID in the States. They reached their endpoints faster than what they thought. So thanks. I'm going to hand over to Lulu.
Thank you, Catherine. That was a wonderful presentation. And thank you for setting the scene. Thank you to the organizers for this opportunity to describe the implementation of this study from a site perspective. And I'm talking on behalf of the Emavundleni site, and it's been a wonderful privilege, an opportunity to be involved in this study and to make a difference to the pandemic and to the lives of people who have been impacted by the pandemic. The results are very, very encouraging and a wonderful triumph for humankind. So I'm just going to give you a brief background about the site I work in and from where we implemented the study. The Emavundleni, or the Emma site, is located in Crossroads. The site was established in 2005, and our site is located within the community. Actually, the neighbors around our site are actually people's homes. So we're very much in the community, and we have expertise in vaccine research, which includes HIV, HPV, TB, and we've also been involved in the evaluation of novel HIV prevention technologies, which include oral products, tablets, injectables, and vaginally administered medical interventions for HIV prevention, in addition to TB and HPV prevention. So this site was one of 4 Desmond Tutu HIV Foundation sites identified to implement the ENSEMBLE study. So right from the outset, when we were preparing to implement the study, we realized, we were told that rapid implementation is vital. And to this end, we put in place several measures. And the first one was to establish an additional site about 700 meters from the main clinical research site. And this was done because we knew we had to see a large number of volunteers to be able to enroll the required number of participants very quickly into the study. We didn't want to interfere with the normal operations of our busy research site, which at that time was implementing 6 clinical trials for HIV and TB prevention and seeing over 1,000 participants. So we established this additional site very close to the main site. And the other reason for establishing this additional site for the ENSEMBLE study was to be able to ensure that we adhered to all the non-medical COVID prevention strategies that we needed to implement. And this included social distancing, controlling the number of participants and staff that we had on site at any given time, ensuring adequate ventilation and no overcrowding, and not putting anyone at any additional risk for COVID infection, considering that we had to enroll individuals at high risk for COVID. We also recruited additional staff to deal with the rapid screening and enrollment. And we had on site a total of 6 doctors and about 8 nurses who were equipped to deal with the study. We extended our working hours. So our normal working hours, like any other research site, is from 8 to 5 o'clock. We had to run clinics from early as 6 in the morning until about 6 to 7 at night, and this included Saturdays. Participants or interested volunteers were presenting to the site as early as 6 o'clock to our wonderful community team, hoping to join the study and make a difference to the COVID epidemic. So long before we started the study, we engaged and sensitized the community through our community team, which is led by a wonderful community liaison officer, Nombeko, who's well known in the community. And community engagement involved engagement with the CAB and other structures within the community. And so we have a very robust community advisory board comprising of 30 individuals. We have an adult CAB and a youth CAB, which is equally split with 15 members in each of these CABs. And our normal approach is to we employ GPP principles, and we discuss the protocol and the informed consent document before any of our studies are implemented. And the CAB is very much engaged with us during the conduct and implementation of the trial, and at the end involved in dissemination of results. So all of our CAB members are from our local communities. They are either community members living in Crossroads, or they are belong to NGOs which work in this community. So we engaged with other stakeholders during this time. We deviated from our standard approach in one-on-one and face-to-face meetings. So we engaged with our stakeholders through WhatsApp groups, which were established at the beginning of the lockdown period. Some of these groups included groups providing education and PPE to the elderly in the community, COVID-19 support groups for people who had already had COVID infection, the local ward counselors in the area, and also some clinic support groups. So in terms of recruitment and knowledge sharing, we normally work in the Nyanga and the Klipfontein districts, which has a population of over 1 million people. But for this study, we expanded our catchment areas because we had to rapidly screen and enroll participants. We do have strong partnerships with local stakeholders, including NGOs, FBOs, and youth groups in the catchment area, and we drew on all of these partnerships to make sure that we rapidly enrolled participants. Nombeko and her team were involved in the sensitization of the community, and this was done through COVID-established WhatsApp groups. Our CLO, Nombeko, has already since the beginning of lockdown, been involved in a number of local radio programs, one in our community here, and then one in the Eastern Cape. So right from the beginning, our organization has been involved in sensitizing the community to COVID vaccines. So now moving on to the results, which Tian briefly touched upon during the at the beginning of this presentation. And so I'm just going to provide a little bit more detail. So in this study, we enrolled globally over 43,000 individuals. And this was in the US, South America, and in South Africa. 15% of this number of participants was from South Africa, and Catherine did indicate that we enrolled over 6,000 participants very rapidly over a period of 1 month. About 33% of the individuals enrolled in the study were older than 60 years and presented with comorbidities. About 41% of the individuals who enrolled in the study had one or more comorbidities, and so that was very important to see what effect the vaccine would have on this group of individuals. So the vaccine, in terms of efficacy, we found that the vaccine was able to prevent up to 66% of mild to moderate infections globally, and in the United States up to 72%, a little bit lower in South Africa, and this was attributed to the variant that we have. More importantly, the vaccine was able to prevent severe infection and hospitalization. 85% of cases were prevented from a severe infection globally, including South Africa. And after 28 days, there were no severe cases in the vaccine groups compared to 5 in the placebo group. In total, there were 5 deaths in the study, and all of these were in the placebo group. So in terms of safety, although we implemented this study very rapidly, and there was an urgency to do that, no one questions that, we all recognize the urgency, safety was paramount, and we followed all the safety protocols that we would normally do for any other protocol, where we would have like up to 2 years to enroll and implement a study. We followed all of the same precautions, reported the same, reported adverse events and serious adverse events in the same manner. And there were no reports of serious side effects in the study, and less than 10% of participants had minor side effects. The advantages of this this vaccine is that there's no cold chain requirement, and so there will be easier access to underserved communities. The vaccine can actually be stored in a fridge for up to 2 to 3 months, and in a freezer for up to 2 years. It's shown to have had a bigger impact on severe illness, and this means lesser strain on our health systems, and that we could get on with the business of looking after people with TB, HIV, diabetes, hypertension, and other comorbidities. Next slide. So Catherine discussed the approval process a little bit more in detail. So we do know that the emergency use authorization would be received in the US probably by the end of February, and other approvals in March and April of this year. There is a move to make available to study participants who receive the placebo to receive active product, and this will be done after the protocol is amended and the study team is working on this. So I'd just like to say thank you to the entire J&J study team, the support that we received from the clinical research organization Acquivia, and all the sites who were involved in the study, including Emavundleni and the other Desmond Tutu Foundation sites. Thank you.
Thank you so much, both Drs. Gill and Nair. Really interesting, really exciting news. Again, congratulations to both of you, the teams, the participants who have made this happen. We also just want to point out that as you ended on a community engagement note, at the outset of even before the launch of this trial, colleagues, we did have a dedicated COVID-19 conversation with Linda Gale Becker. We also did a podcast, a range of frequently asked questions, Q&As, as well as an op-ed, all prior to this trial. So this has been one of the few trials that did consciously reach out, engage with civil society, and really practice an open door policy and a willingness to be open and transparent as far as they could be. So a recognition and thank you for that. But I think we have quite a few questions that have already come through, so we will jump right in. A reminder, please share your questions and comments in the chat box. To access all of the previous recordings and materials, we will put a link into the chat box, but all of that is on our website, africanalliance.org.za. So some of the initial questions are really around community engagement. I think you've laid out the organizations and advocates and movements that you engaged with. Could you perhaps share what you have learned in this particular community engagement process for this trial? How different did community engagement look, feel, and manifest in this instance, given the conditions? We're told it's an emergency, we needed answers quickly, there was huge pressure on researchers, on regulators, on everyone really, to get to answers quicker. What were some of the challenges in maintaining ethical considerations, especially around recruiting? And how was that received by the community, given the time pressures?
Okay, okay, you go, Lulu.
Okay, thank you. So I think for us, the big challenge was that we couldn't employ our tried and tested methods of engaging with the community and recruiting participants because we now were working under lockdown conditions, and more, rather than engaging through face-to-face meetings and going out into the community, meeting stakeholders, sitting down, discussing the study, and meeting one-on-one with participants, door-to-door recruitment, meeting in schools and other community locations. We had to look at electronic platforms, look at WhatsApp, and I think that was a challenge also because we had to reach out to groups who normally don't use social platforms to engage on, and so that was a little bit of a challenge, reaching out to older participants. But the community team and were like really engaged, sat down with people, got them onto platforms, WhatsApp, taught them how to use emails and that kind of thing, because that was very important after after recruitment and being enrolled in the study as well. Yeah, that's from the Emma perspective, and over to you, Catherine.
Yeah, so I think very similar from Masi. For us, so there were two things that were very different about this study. The one was the really quick timeframes. I mean, to recruit all of your participants, for us, we only started at the end of November, so we had about 2 or 3 weeks to recruit all of our participants. So that for us is very different, and I think often the engagement piece is sacrificed when you move faster. So as Lulu said, we did have to be creative. The other thing that was quite new for us was it was a different kind of person that we were recruiting. We were going specifically for for older people, often quite difficult to engage them on social media. So we really had to think carefully. I think what made the study easier is that people tend to, in our area, tend to believe in vaccines, and a vaccine is quite it's quite an easy thing to present to somebody, because people know that children are vaccinated against diseases. So for us, it wasn't difficult at all to recruit people. People were very keen, and I think people's general literacy around COVID is quite high. People have had a year to kind of absorb what COVID means, and I think with the lockdowns, and I think people also get the severity of this disease. Mm-hmm. So we didn't have any issues recruiting. There were long lines of people waiting to come and join. I think the other thing that people have appreciated is that it might be a long time before a vaccine is actually widely available, and this was a way to get access to a vaccine earlier.
Yeah, no, thank you for that. The next question we have is, how were these clinical trial sites chosen globally, and then in particular South Africa? Could you just give some a brief overview of that? How were the sites chosen?
So I think go ahead.
Go, Lily.
Okay, so I think it's based on the past experience of the clinical trial sites, and also so we were recruited through a network that we work with already, the HIV Vaccine Trials Network, because having had experience with implementing HIV vaccine and TB trials in the past. And so I think it was, looking at what infrastructure the sites had in place, their history and success with recruiting in previous trials, and how quickly we could do that. We had to say that we would be able to, we would have sufficient space to implement the study. We have the catchment population to work with. And I think all of those things were taken into account. And we did have to provide reassurance that we could do this very quickly.
Yeah. And I think that in part speaks to the long history and legacy of the foundation's work in those communities, in terms of trust and proximity to communities. Dr. Gill, perhaps if I could pass this one on to you. In October, we hadn't really understood, or to a large extent even heard, of a COVID variant. Now that we do, what do these interim results mean for the variant discussion? Do they mean anything? Does it apply across the board irrespective of what variant comes up?
Sorry, was that to me or to Lulu?
It was to you or Lulu, whoever's comfortable answering it. It was really around the variant.
Okay, so I think with the variants, there is a very specific variant in South Africa. What's really nice about the study is that we know now specifically how well the J&J vaccine works against that variant. It's the same with the Novavax study. We now have actual data on how it works. I think the thinking is that the virus will continue to mutate. There are already 3 named variants. So I think I think that it will carry on mutating. And I think the drug companies are going to have to tweak the vaccine like they do with the flu vaccine every, I don't know, maybe every 6 months, every year, to make sure that it's active against whatever variant is prevalent at the time. I have heard data from Moderna that they don't think that this is going to be too difficult, but it will require some sort of surveillance, sort of ongoing surveillance to see which variants are more prevalent in which areas, and then getting a vaccine that can work against those variants. I think the other thing to remember is that despite the different variants in the different countries, the J&J vaccine could prevent death and severe illness across all the variants, and that's something that is a definite take-home message. So while it didn't work as well against the mild version of our variant, it worked just as well against to prevent death and serious illness. And the scientists are unpacking that, because it is an interesting idea that it didn't prevent mild illness, but it could prevent severe illness and death. But I think that's the key take home, that it can prevent death and severe illness across all the variants seen in the study.
Thanks for that. Kinelo is asking about severe adverse events. Did you see any? Could you give a summary or a snapshot of what those were, if there were any, and how were those managed?
So I think Lulu actually presented that in her presentation. There weren't any severe adverse events seen. I mean, I know for my site, we definitely didn't have any. And actually, what we did see was that not many of our participants had any kind of reaction to the vaccine. So what we always do on vaccine studies is, after you've given the vaccine, you observe your participant for half an hour after the vaccine, just to make sure that they're okay. And we didn't see anything. I'm not sure about Lulu, and afterwards, nothing as well. It seems to be very well tolerated.
I have to agree with Catherine. Actually, we didn't see any adverse events at all. The things we normally see, like a localized reaction to the injection, redness, swelling, and so on. We saw nothing of that sort, and it was quite surprising. And like, yeah, we had to we actually questioned whether, people were actually reporting accurately, because we were so surprised that we were not seeing any of these. But we have a mechanism in place to follow up with participants and ensure that they are reporting in a timely fashion.
Yeah, yeah. Thanks for that. Another question which perhaps you could help us understand a bit better, that might or might not be directly related to these results, is what do we make of the people who have already been infected? And what do we now know? What is the latest on immunity? If those previously exposed people are still immune, then that's a big step towards reaching herd immunity. The hypothesis is that people who have had severe disease are probably better protected from being reinfected. Do we know how long immunity would last in someone who had a symptomatic infection versus those who have had severe disease? And do these results mean anything for that immunity discussion?
I think we don't know that answer about how long immunity to natural infection lasts. And we are currently involved in a study where and Catherine as well, we've enrolled participants who have, or patients who've had this who've been infected, and we are following them up for a period of over a year to determine how long immunity lasts. So we're seeing them periodically, collecting swabs and bloods to try to ascertain how long immunity via natural infection is conferred.
Thanks for that. I think sorry, Dr. Gill, would you like to add something?
Yeah, so I think what is interesting about the J&J study is that they actually did enroll people who had had previous COVID. I mean, it wasn't, we didn't necessarily ask people, but it wasn't an exclusion like it is on some of the other studies. That is a secondary objective. So they're only going to have a look at that maybe after a year, after 2 years, to try and see, so people who had antibodies at baseline, whether they, are reinfected. So that's a much longer analysis. I don't think you won't get that in the first month. So I think the data on that is coming, and it will be really interesting. And I'm glad we didn't exclude people who've had COVID, especially now after the second wave. And a lot of us have now had COVID and are wondering whether we should actually get vaccinated.
Mm-hmm. I think the discussion has certainly been increasingly around access. And what does all of this mean? What do all of these exciting results and pioneering science and just the sheer volume of investment, thought leadership, community engagement, community ownership that is being put into getting these vaccines? What does it all mean, for example, when at the end of the day, communities just won't have access? One of the questions that we've shared is, why should communities agree to participate in vaccine trials going forward if they're not going to have access to those vaccines? And I know that's not your specific area, but it's something that we really try to ask a lot of our research leaders because of the work and the effort that's being put into this vaccine. What is your thoughts on eventual access and when this vaccine will actually end up in the bodies of those who want it and those who need it? What are your thoughts on the delays in getting vaccines, irrespective of which vaccine, to communities who need it?
Yeah, so I think I'll take that. I think just to say up front, it is just as frustrating for the answer to this is that it's just as frustrating for researchers. We're also feeling, angry and frustrated that our sites have participated in these studies and that we don't have access. So we're very much on the same page. I think what these vaccine studies mean, and why I was, praying that this vaccine would work, is that it just gives us a lot more options. We don't have to rely on one drug company who would then have a monopoly and could charge as much money as they wanted to. So I think if a lot of these vaccines work, it will make access faster. So I think that's the good thing. If we've got 10 and all 10 are working, we will get access quicker. And I think because we're making the vaccine in this country, it's more likely that we'll get access to the J&J. I also think that these results are good news. As Lulu said, the vaccine can prevent severe illness and it can prevent death. And that for South Africa is going to be a game changer. It's going to keep people out of hospitals. Our hospitals already got, 100% bed occupancy. And I think getting a vaccine out quickly will be a game changer for South Africa, and especially a one shot that doesn't need a rigorous cold chain. So I think for me, the J&J results are great. Access is always difficult, and I think that it might need, it's going to need a lot of activism to keep the government on track. Probably Absolutely. Similar to what we saw in the early days of HIV and antiretroviral access.
And I guess even more recently, looking at the Vida vaccine, the assumption was that these vaccines are being trialed in South Africa. The research is being made possible by people in South Africa. The research is partially funded by the public purse. And so the argument or the idea that's put forward is that we've made all of these investments will result in early access. I think we've seen that repeatedly come crashing down to nothing. And so it's really interesting to see how ENSEMBLE will navigate those waters going forward. And I guess more broadly, what does this all mean for the next vaccine trial? So according to some of the latest thinking, general public is only going to access this vaccine perhaps the second quarter of next year. So with such a lag, what does that do to communities' excitement, willingness to hear and to engage in the range of other vaccine trials going forward? So I think it's a really interesting space. And certainly it's something that we as a collective have to keep on the agenda. I know we're 8 minutes to the top of the hour. One question which...
Yeah, can I just make a comment? I think it's wonderful that the study participants will get the vaccine first, that those who were on placebo will have access as soon as the protocol can be amended. And then the other thing is, I think providing the vaccine through a central mechanism, through one central mechanism, is an excellent idea, because it will ensure equitable access. No matter who you are, you will stand in the queue for the vaccine, and it will be rolled out according to your risk. And I think that's something that's a good thing.
Yeah, absolutely. And we'll certainly be keeping an eye on that to make sure it happens. I think a last closing question, which I'll open. I know it's not something directly related, but just to say that, with the current news cycle highlighting the fact that manufacturers are insisting on indemnity from any kind of liability on their vaccines, this really sets back the work, doesn't it? Because the common understanding would be, well, what are they worried about if the vaccines are safe and they work well? Why would a condition of procurement, especially through bodies like COVAX, come with those indemnity from any liability of the product? So it's really, really an interesting time, I think, to have a look at this. And I know that certainly in the last year that we've been engaging around COVID, it's really made us think deeper and ask some of the harder questions in terms of the role of researchers and scientists in the bigger picture. You know, frequently researchers or scientists will tell us, you know, our job is the science and we don't get involved in the politics of pharma or access or distribution. But what is personally encouraging to me is that we're increasingly seeing researchers and scientists like yourselves who are really having clarity of thought in terms of the investment you're making. All of this investment in time and money has to result in something. And that something has to be access. And that access has to be equitable. So I know we're 5 minutes to the hour. I will just go and see if we have any questions in the chat box that we have not gotten to. It appears you have covered most of them. Are there any final words, any comments? One lesson learned from both of you? Would you do anything different if there were no time pressures going ahead? If we could have one lesson learned or one reflection from both of you, specifically around Ensemble. What is your takeaway message? What would you like to leave us with to think about with regards to these interim study results? We'll start with Dr. Gill.
Yeah, so I think my take home was having, I think for myself as a researcher, I naively thought that if we did participate in the J&J vaccine, that we would automatically get it. And I think that was, you know, if I look back on that thinking now, it is naive. And I think going into you asked what would be different if we did a new vaccine. I think going into a new vaccine with more terms and conditions, you know, we have amazing infrastructure in this country to conduct clinical trials. And I think we need to be better at negotiating access upfront. So I think that would be my learning point, that you can't assume that access will happen, especially when you're working with pharma.
Absolutely. Dr. Nair?
Yes, so I think, you know, when we think about implementing our trials and we do our we compile our informed consent documents and make our ethics submissions, and we always include a statement that about post-trial access, and we really do think about post-trial access, and we discuss this with our communities when we do community engagement before the trial starts. And I think maybe just, you know, we need to do more than just having that one line saying, you know, there will be post-trial access and placebo recipients will receive the active product. I think we need to think more and maybe have a stronger statement regarding benefit sharing in our informed consent documents and when we engage with our communities.
Absolutely. Thank you so much for that. One last comment, which I won't ask you to address, but just put out there, certainly raised in terms of the specific role of pharma in this pandemic and how that has almost altered and colored and shaped what the research looked like. As opposed to other research where perhaps pharma didn't play such a dominant and certainly public role. So something for us to reflect on and think. But Drs. Gill and Nair, thank you so much for joining us. It's been an absolute pleasure speaking. And as I had said to our participants, we will, by tomorrow, both the recording, the transcript, and the Q&A will be on africanalliance.org.za, as well as our YouTube channel. And over the coming weeks, we will be releasing an opinion piece, as well as frequently asked questions guide. But really exciting times. Next week, we speak to Professor Graham Menkes and Principal Investigator Amy Ward on a new phase 1 vaccine trial that has just been approved by UCT Ethics and the national regulator SAPRA and is planned to kick off in Khayelitsha. This trial will use a second-generation human adenovirus 5, Ad5, that was initially designed for cancer therapeutic vaccines, but has now been repurposed for SARS-CoV-2. A phase 1 trial has been completed in the US, and good safety has been shown. The trial will be repeated in South Africa with a similar design, but colleagues will remember that that special attention, and we need to pay special attention to this particular trial. You will remember the Ad5 discussions from STEP and Pambili that specifically raised issues around increased HIV acquisition in men with the Ad5 vaccine candidates. And so Professor Menkes and Amy Ward will explain to us what the fundamental difference in this trial is. We will also unpack with them the informed consent process, what they will be offering in terms of HIV prevention services, including referral for PrEP and a range of other issues. So really, really exciting, the return of HAD5 to South Africa. What does this mean on the back of STEP and Pambili? But again, thank you so much everyone for joining us on this Thursday morning. Next week, Thursday, we look forward to seeing you again at 10 o'clock, and we wish all the best to the Ensemble team in your ongoing work, and look forward to access. Thank you so much for joining us, and have a great Thursday.
Thank you, Tian.
Bye.
Bye-bye.